things. Okay, go ahead.
Okay, so it is my absolute great pleasure to introduce Alison Feder today. So usually when we introduce seminar speakers, we don't need to go way back to her undergraduate years to sort of explain things. But in Alison's case, she was producing top quality research at this stage. So this is where I'll begin. Alison did her undergrad work in mathematics at the University of Pennsylvania, where she was advised by Warren Ewens of the Ewan sampling formula and Joshua Plotkin. During this time, she published a lovely genetics paper on identifying signatures of selection in temporal data. During summers of her undergraduate years, Alison traveled west, where she worked with Rasmus Nielsen and Monty Slatkin at UC Berkeley, and then later Dmitry Petrov at Stanford. After undergrad, she got a master's in stats at Oxford, advised by Gil McVean. Then she did her PhD with Dmitry Petrov, collaborating with Pluni Pennings. During this time, she did her work on the evolution of drug resistance in HIV. In particular, Alison explored how resistant alleles spread in HIV populations within a patient, showing that the efficacy of drugs changes the nature of selective sweeps. Her eLife paper on this is one of my favorite papers. I just think it makes a great undergrad evolution paper. You should use it in courses. And currently, Alison is a Miller Fellow postdoc at UC Berkeley and an incoming faculty member at UW Genome Sciences in the summer of this year, if I'm correct. So with that, I will hand it over to Alison, if you want to share your screen.
Yeah, thanks, Vince. That is a very nice introduction. Got to press the button, got to see if I can figure out how to do this again. But yeah, thank you all so much for having me here. I'm excited to be virtually visiting the Institute, and I hope that I can come back someday in person. Everyone can see my screen okay? Cool. Because now, yeah, I'm in the neighborhood and I would love to help bridge the communities in Washington and Oregon. And I think that there are a lot of great synergies that have yet to, well, that are already being exploited, but that there's always room to add more. So as Vince said, my name is Alison Feder. I'm a postdoc at Berkeley, and today I'm going to be telling you about some of the work I've done on evolutionary dynamics in HIV intra-patient populations. And I'm really interested in populations that evolve really quickly and the sorts of ways that you need to change your thinking when you're considering these types of populations. And so today I'm going to be using HIV as an example of these types of populations. I'm going to start by telling you how quickly HIV evolves drug resistance, how this has changed over the course of the epidemic as our drugs have gotten better and better, how HIV continues to evolve resistance despite the fact that our drugs have gotten so much better, and then finally how spatial structure may enable or what it looks like when HIV is actually evolving within a person with respect to space. So to start off, for a long time, we thought that evolution was this slow and gradual process, but we're increasingly realizing that in a wide variety of natural populations, evolution can actually be extremely rapid. And although these populations create some of the world's most troubling medical and agricultural problems, there's still a lot we don't understand about how adaptation proceeds in this kind of fundamentally important regime. And so as I mentioned, I'm going to be using HIV to illustrate how you kind of need to change your perspective when you consider populations such as this. And I want to start out with just a concrete example of how fast HIV evolves drug resistance de novo within its patients. So we can consider the population of viruses inside of a completely drug-naive patient. Oh,
Alison, yeah. Real quick, you've got a Logitech presentation update window you might
Gotcha, yeah, I see it. I think I need to close out really quickly in order to do that. Yeah, thanks for the heads up.
Okay.
Let me try this again. Great. Cool. View.
Sorry to get in the way of your momentum, but it's all good.
No, it's totally great. And if there are questions that come up during the talk, I should mention, feel free to interrupt, especially if it's something that impedes your understanding, like a Logitech banner. So yeah, HIV evolves really quickly. We're going to look at a particular time course in a particular patient. So we can assay the virus for large effect drug resistance mutations that are easy to sample from the blood and ask how quickly the population composition changes. And so what we see is drug is starting to apply at week 0, and then really quickly, we can see that within this patient, a drug resistance mutation goes from effectively 0% frequency to nearly 100% frequency in something like 4 weeks. And this, looking at a graph like this, it sort of begs the question, how is it possible that evolution is happening so quickly? And it's actually not just this patient in which drug resistance happens so quickly. So I'm going to show you 18 additional patients. And what we see is that across a wide variety of these patients, drug resistance happens this quickly in this repeatable fashion. So 80% of the populations in this instance for patients, I think I failed to mention, these are patients who were treated with 3TC monotherapy in 1995 or slightly preceding 1995, we see that 80% of the populations have resistance in something like 30 generations, with potentially more individuals evolving shortly thereafter. So the question then becomes, how can we explain evolution that's not just fast, but also is predictable? And to do this, we go back to sort of the simplest case that we could imagine of a model of evolution, which is to say the Wright-Fisher model with selection. So we could imagine that we have a population of N viruses, and each generation, each virus has a probability mu of acquiring a drug resistance mutation. This joint parameter N times mu, which I'll also call theta throughout the talk, is the population mutation rate and is just telling us how many of these drug resistance mutations are entering the population each generation. And when you have one of these drug resistance mutation, it confers a selective benefit 1 plus S relative to the wild type that doesn't have the mutation. So in order to try to understand how it's possible that evolution happens so quickly and predictably, we can think about a range of values for selection strengths and population mutation rates and just ask, if you do forward simulations, how likely is it that you're going to get a selective sweep, which is to say maybe a drug resistance mutation getting higher than 50% frequency in 30 generations. So this is something that we recently did, and it turns out that if you do these experiments, you find that you need to have a very constrained part of the parameter space, or a relatively small part of the parameter space, where this is actually possible to do. So namely, your selection strength has to be quite strong, otherwise sweeps aren't going to happen fast enough. But also your population mutation rate has to be pretty high. Otherwise, you aren't going to have enough beneficial mutations entering the population that are available to actually do the sweep in 30 generations. So I want to sort of draw your attention to the scope of or the magnitude of the parameters that are present here. So if we think about the strongest examples of human adaptation, things like lactase persistence that we know are really strongly positively selected in humans, these are sort of on the values of like 0.01 to 0.05 is what people estimate. And so we're thinking about things in a part of the parameter regime that is way higher, way more extreme than what we would generally think about with humans. So during the talk today, I'm going to kind of be taking us into this regime of parameter space and asking, what do things look like when we live up here? So the first question that we'll try to answer today is, what does evolution look like under these strong population genetic forces? What are the actual patterns that we would expect in how alleles spread? Second, we're going to ask how reducing the rate of evolution has changed the dynamics of resistance emergence. So over time, we've become better and better at treating HIV. It's not the standard case that now you give someone 3TC and they evolve drug resistance in 4 weeks. By changing the rates of evolution, we might have some predictions about how the dynamics of evolution could be changing as well. Despite the fact that we're much better at treating HIV now, evolution is still able to happen in HIV, and there are patients who still become drug resistant despite the fact that we're treating them with these good combination therapies. So we're going to ask, how can evolution overcome these complex simultaneous challenges that we apply when we give patients multiple combinations of drugs? And finally, we're going to think about how the strong evolutionary forces that we've talked about in the first 3 points are going to play out in spatial contexts, because I think this fourth point actually has a lot to do with the third point. But I want to get started on the first one. Just what does evolution look like when popgen's forces are really strong? And to do this, it helps to return to a classical picture of what we think about how adaptation happens in most populations. So if you have an allele that is going to sweep in the population, after some maybe change in the population conditions, you first have some waiting time for a beneficial mutation that is proportional to the inverse of the population mutation rate. So the more mutations entering the population, the less time you have to wait. A beneficial mutation appears and then begins to spread within the population. This is our very classical hard selective sweep. And it's sort of what many people would consider if they're thinking about how adaptation happens in wild or experimental populations. But I want to remind you that we're actually going back to this part of the parameter regime where the population mutation rate is really big and the selection strength is really strong. So if we crank up the parameters that were in this regime of the parameter space, we see that the waiting time shrinks significantly because we're thinking about large values for theta. And as before, we may have a beneficial mutation that enters the population and begins to spread, but actually the sweep time for that beneficial mutation to go from low frequency to high frequency is longer than the waiting time for a new beneficial mutation to enter the population. So we might expect that before that sweep finishes, a second beneficial mutation might enter the population and begin to spread in parallel. Now, this is an example of what we would call a soft selective sweep. And soft selective sweeps have sort of been a subject of some interest since, I guess, the early 2010s when Pennings and Hermison put out several papers on the subject. Sorry, maybe even earlier than that. I'm not Vince. I don't know my dates perfectly of all of these papers. So in the case of, it turns out that actually knowing something about how quickly these sweeps are happening can tell us something about the manner of the way that these sweeps are happening. Namely, that if we're thinking about the parts of the parameter space in which we're likely to have sweeps in 30 generations, it turns out that these are exactly the parts of the parameter space in which those sweeps are likely to be soft. And in part, this makes sense because the fact that you have a soft selective sweep happening within a patient is indicative that the probability of a beneficial mutation being produced is so high that it actually is happening multiple times within the same patient. And so thus it's sort of unsurprising that we might see a beneficial mutation happening across many different types of patients. So what this leads us to believe is that at least early in the HIV epidemic, there was probably at least some prevalence of soft selective sweeps in driving the evolution of drug resistance. And this is something that we can actually empirically see in clinical data if we actually just go down to the sequencing data. And I'm going to show you one particularly nice example of this. So there's a particular mutation, it's called K103N. It's a lysine to asparagine position at the 103rd position of reverse transcriptase, which is sort of one crucial gene that is targeted in HIV evolution. Or in HIV, when one of the drugs that we have target this reverse transcriptase. And so lysine is encoded by AAA or AAG, whereas asparagine is encoded by AAT or AAC. So what this means is that there are two semi-equivalent ways to encode asparagine, and that changing an A to a T or an A to a C at the third position sort of equivalently creates this K103N mutation. So what this means is that if we were to look into clinical data and we see the co-occurrence of AAT and AAC, this would point to the fact that at least two beneficial mutations must have entered the population in order to create this pattern. And so some of my colleagues, including Clooney Pennings, actually did this in 2014, where they looked at a patient that at the, right before they started treatment, you couldn't see anything at position 103. So here you can see that everything is AAA or lysine, but they measured this patient, or they examined sequences from this patient in time series. And at a later time, day 28, we see that there are both AAT and AAC mutations that are circulating. So even though if you were assaying this at the amino acid level, you would say, oh, well, you've got a K103N mutation that is fixed in the population. If you look under the hood at the nucleotide level, you can see that this sweep was actually soft. So what we see is that in the early days of HIV, it seems like soft sweeps were an important driver of drug resistance in patients who were treated with the types of therapies that we were giving patients in 1995. But over the past years, we've gotten much better at treating HIV. And now, instead of just giving patients one drug at a time, we give them combinations of multiple different types of drugs that target different stages of the viral life cycle. And so what this means is that single drug resistance mutations are normally, we would consider insufficient in order to provide full drug resistance to all of the therapies that are present. And not only did we learn how to combine these drugs in different ways, but we also just created more and more types of drugs. And as a result, the incidence of drug resistance fell drastically over the past, several decades. And it's really an amazing triumph of modern medicine, but it's also something that we can try to learn about. So we might have some reasoning to believe that as the drugs have gotten better, that there's been a transition away from soft selective sweeps. And I'll show you why I believe this in a second. But second, what we're going to do is we're going to ask how reducing the rate of evolution has changed the dynamics of resistance emergence. And as I already sort of alluded to, we think these things should be connected because the probability of mutations happening multiple times within the same patient may also be related to across patients how likely sweeps are to happen, independently in multiple patients. So over the past several decades, we've gone from this sort of bluish area up here where resistance was really likely to happen over very short timescales into this red area over here where resistance became much less likely. So how, in terms of movement on this graph, did that happen? Well, one hypothesis is that as drugs have gotten better, selection has become a weaker force. So sweeps have sort of transited more slowly from low frequency to high frequency. And so the reason that we see fewer sweeps in 30 generations is just because the sweeps take longer. Now, there's not a lot of evidence for this hypothesis, namely because observing drugs at intermediate frequencies is really rare, regardless of what type of therapy you give. It just basically means that all sweeps are happening pretty quickly. Another hypothesis is that maybe over time, what we've done as we've gotten better drugs is we've decreased the population mutation rate. And so we've gone sort of in this direction. Now, if this is the case, if we've gone from here to here by decreasing the population mutation rate with these better drugs, it's possible that at the same time, we've decreased the probability of having soft selective sweeps. We've effectively moved from this area where soft sweeps are common to one in which soft sweeps are increasingly rare. So this is a hypothesis that we're going to try to test. Is it the case that as our drugs have gotten better, that we've decreased the probability of soft selective sweeps and increased the probability of hard selective sweeps in the relatively rare cases in which drug resistance does emerge? So sort of just to say that again, when evolution of resistance was relatively common, we have some evidence already that it was driven by soft sweeps. So what's happening down here when sweeps have become rare? Have we seen this transition to hard selective sweeps? And so to try to answer this question, I'm going to show you data from so to think about how to answer this question, we need to think about how we can tell hard and soft sweeps apart in genetic data. So in the case of a hard selective sweep, because a beneficial mutation lands on one genetic background that increases the high frequency, much diversity is lost as the sweep happens. This is sort of our classical signature of a hard selective sweep, is that this depression of diversity surrounding the sweeping allele. But in the case of a soft selective sweep, because beneficial mutations land on multiple different genetic backgrounds that may have some accumulated genetic diversity already, much more diversity is retained following a soft selective sweep, simply because more variation is kept within the population, even as there may be fixation at a particular site. So what we can do is look for different signatures of diversity following selective sweeps in order to try to disentangle these two hypotheses. And we're going to do this in patients who are treated with various different types of HIV drugs so we can compare early and late pandemic or epidemic drugs. So to do this, we looked at 7,000 patients approximately from the Stanford HIV drug resistance database that were collected between, I think the earliest one was in 1989 and the latest was in 2013. For each of these patients, we know what drug that patient was given. It could be a combination therapy of multiple different drugs, like a modern therapy. However, we know that they were treated only with exactly one drug therapy, so it could be 3 drugs. We also have a single HIV consensus sequence from each patient. And this is great because we can have, one of the nice things about working in an HIV system is that there's a really well-defined genotype to phenotype map. So if you give me a sequence and you tell me what drug the patient is receiving, I can basically tell you which of these particular mutations or which of these genetic positions are drug resistance mutations and have some sense of what the phenotype of the HIV virus is. But there's a problem here, which is that I've told you that we have only a single consensus sequence per individual. And that's problematic because I've just told you that we're going to try to look for changes in diversity signatures in order to look for hard and soft selective sweeps. Fortunately, we can get around this to some extent by focusing in on the exact type of data that we're using, which is largely Sanger sequences taken from patients dating back to the early 90s largely. And in these Sanger sequences, what you're often left with is a chromatogram that describes the intensity of the support for particular nucleotide positions or particular nucleotides at each position. And in some instances on this chromatogram, you have places in which there's support for multiple different nucleotides at a particular position. Here you can see it in this sort of S and A, which could point to either a G or a C at this position. Now, ordinarily, that may be kind of annoying if it means that you don't have clear resolution on what exactly this nucleotide is at this position. But actually, in the case of these large, diverse viral populations within patients, there's good reason to believe that there actually is underlying diversity that is creating this CG dimorphism. So in this case, what we're going to be doing is using mixture nucleotide calls as a sort of proxy for multiple circulating types within a certain patient. And this is relatively well supported because you can go back and sort of validate this measure by asking in a different data set in which you have a much higher resolution data set of multiple sequences taken from the same patient. You can see which are the sort of sites that are diverse within a patient on more traditional metrics such as pi, and compare them to the proportion of ambiguous reads across patients at that same position. What you see is that the positions that tend to be ambiguous across patients tend to be diverse by more traditional metrics within patients. So we felt pretty good about using this as a sort of proxy for whether sequences were diverse or not. So the idea being that the more ambiguous reads you have in a sequence, the more nucleotide diversity you likely have in that patient. So what this means is that for each of these individual patients, we know we can measure how many drug resistance mutations they have, and we have some metric of diversity is defined by ambiguous reads. Now, this still sort of presents a problem, because what I've told you is that in order to look for the differential signatures of hard and soft selective sweeps, you need to look at post-sweep diversity signatures, and we don't have any time series data. We just have a single time point per patient. But fortunately, what we do have is many patients that were treated with a wide variety of different drugs. And so what we can do is think about a particular treatment of interest and then just isolate down to all the patients who were given this particular treatment, and then organize them. Sorry, this slide always lags a little bit because all of the moving people. And organize them based on the number of drug resistance mutations they have. So what you can do is compare patients that don't have any drug resistance mutations to those that have one, that have two. And we would expect that if these patients are undergoing hard or soft selective sweeps, they should have a differential relationship between the number of drug resistance mutations they have and the effect of fixing a drug resistance mutation on diversity. So this is the model that we're trying to fit effectively, is the amount of diversity in given a treatment based on the number of sweeps that have happened within that patient. Now, because we can't measure these things directly, what we actually measure in the data is the number of ambiguous calls as a function of the number of drug resistance mutations that a particular individual observation has. And this isn't a perfect proxy because, you could potentially have a sweep that has multiple different drug resistance mutation, but it kind of gets at the core of the question: what is the relationship between fixing a drug resistance mutation and the effect in genetic diversity? So this is how I'm going to show you the data. Is basically on the x-axis we have treatment efficacy, and I'm going to be showing you a wide variety of different treatments that are going to sort of fall somewhere in this range. If they're over here, they're going to be treatments that almost never fail. If they're over here, they're going to be treatments that almost always fail within a year. So treatment efficacy here is defined as the percentage of patients that have suppressed viral loads after 52 weeks. And on the y-axis, we have this term from our regression, which is the change in diversity associated with fixing a drug resistance mutation, with the idea being that the more negative this term is, the larger the decrease in diversity associated with fixing a drug resistance mutation. And I'm going to start by showing you a single therapy. This is 3TC plus AZT. It's a 2-drug combination therapy where both of the drugs are the same class. It's not very good. It doesn't work very often. And what we see is actually it's associated with a slight increase in diversity associated with fixing a drug resistance mutation. So far from decreasing diversity as we might expect in a hard selective sweep, it appears to be slightly increasing in diversity. And now I'm going to show you all the other treatments that had enough data that we could fit them within our model. What we see is this negative relationship between the amount of genetic diversity associated with fixing a drug resistance mutation and the efficacy of the treatment. Furthermore, this is a strong negative relationship, and that's even accounting for, even accounting for these relatively high outliers up here. This is AZT. This is like the very first HIV drug that failed almost immediately in most patients, very much associated with soft selective sweeps, we think. Furthermore, if you color these different therapies based on the year in which the drugs were, the therapies were given, we can see that the most modern therapies are associated with the largest decreases in diversity associated with fixing a drug resistance mutation. So what we think this means is that this sort of confirms what we had already seen previously, which is that there's this positive or non-negative relationship between the amount of diversity or the change in diversity associated with fixing a drug resistance mutation earlier in the pandemic. And this is sort of corroborated with what we understand from the time series of these sweeps and from the patterns that we can see in haplotype data. But as drugs have improved, we seem to have pushed HIV into this part of the parameter space where hard sweeps have become the predominant mode of adaptation. So reducing the rate of evolution has changed the dynamics of resistance emergence via this transition to hard selective sweeps, as we may have imagined it would. And what I find really exciting about this is that it connects the dynamics of intra-patient evolution to the rate of evolution across a variety of patients. So what this means is that hypothetically, if you were to give me a drug that you know nothing about, and you're only able to give this drug to one patient, and this patient comes back a year later and they've become drug resistant to this particular drug, by looking at the amount of diversity within the population, you might be able to tell something about how well this particular drug might work across a variety of patients. So for example, if this patient had failed via a soft selective sweep, that would mean that resistance evolution was so likely to happen within this patient that it happened multiple times. Whereas if this patient comes back and they had sort of undergone a hard selective sweep of drug resistance, this might point to the fact that this patient was somewhat unlucky, and it doesn't necessarily necessitate that this drug is not going to work in other patients for extended periods of time. Okay, so what we've seen is that by studying this transition from bad drugs to good drugs, we have this transition in how evolution is happening. But I kind of skimmed over something previously, which is how exactly is it the case that these combination therapies of multiple drugs that we think work really well are failing at all? So the reason that we think that these drugs work so well, or the sort of way that I've illustrated drug resistance evolution so far, is that you have a particular virus and you treat it with a particular drug, and the drug stops the virus from being able to replicate. Now, with a certain probability, the virus can become drug resistant, and then the drug stops working on the virus. Now, the idea behind these combination therapies is that you have a virus again, but you treat it with 3 drugs simultaneously. And each of these 3 drugs are independently inhibiting the virus. So even if the virus gets a single drug resistance mutation, it only confers resistance to 1 drug, and the effective R naught of the virus within a patient is still below 1, and the viral population size decreases, and the particular mutation isn't able to spread. Now, what needs to happen in order for a virus to be resistant to all 3 drugs simultaneously is this much higher order mutation. Maybe within a single round of replication, it needs to get all 3 mutations simultaneously in order for the virus to be able to spread in the presence of the drugs. And this is sort of the reasoning behind why we think these drugs work so well. And it's become kind of such a classic example that it has made it into courses about why we need to teach evolution to medical students. It's become a sort of textbook example of how evolutionary thinking has been able to prevent the evolution of resistance. And so because it's a textbook example, I'm just going to read you a quote from a textbook. Combination therapy has shown great success in the context of HIV infection. In a typical HIV infected patient, we can expect that viruses resistant to 1 or 2 drugs are present. However, it is extremely unlikely that a virus resistant to 3 drugs exists. This provides the rationale for why a combination of 3 drugs is required to achieve long-term suppression of the virus by drug therapy. So what this means, or so this is sort of, as I mentioned, this lauded example of why evolutionary thinking is so important. But I want to argue that actually things are a little bit more complicated than how they've been laid out here. So this is a graph that I've shown you previously, describes the rate of evolution of resistance and how it's decreased as we've gotten better and better drugs. But I want to note that the first HIV combination therapy was introduced in the mid-90s. So a lot of these high levels of drug resistance evolution were actually ongoing while we've had relatively good combination therapies. So it seems like there's something missing in this explanation that can't necessarily explain why we've had so much more evolution of resistance, despite the fact that we have been treating patients with 3 drugs for a long period of time. So in this sort of third part of the talk, I want to address this question of how is it that evolution overcomes these complex simultaneous challenges of being treated with multiple drugs simultaneously. So maybe the idea behind this model is still correct. Our internal picture that we need 3 single drug resistance mutations on the same virus in order to spread is correct, but the idea of the rates of how often this happens is sort of not quite right. And to do this, we can look at patients that have been treated with combination therapies and look at the patterns through which the patterns of drug resistance emerge in these types of patients. So we would imagine that under this model that was laid out in this textbook, that if you treat patients with 3 drugs, almost all of them are going to have no drug resistance mutations. But among the patients that do have drug resistance, they should live down here in this triple combination therapy or triple resistance category, where they're resistant to the blue drug and the yellow drug and the light blue drug. And so maybe we have some sort of flawed idea of how quickly patients are transiting from this top category to this bottom category, but in general, the model is correct. And we can actually check this with data. So you can plug in a particular combination therapy here, 3TC, NFV, and D4T, which is a particular therapy that was tested out in the early 2000s. And what we see is that, sure enough, in accordance with the model, many patients live up here having no drug resistance mutations. But actually, it isn't the case that most of the patients are living down here in this triple drug resistance category. We see patients that have single drug resistance and double drug resistance and triple drug resistance. So it does seem like there are situations in which resistance to a single drug at a time actually is sufficient in order to allow drug resistance mutations to spread within a patient. Furthermore, it seems like they're following these sort of semi-predictable patterns that allow patients to sort of stepping stone from resistance to 1 drug to 2 drugs to 3 drugs. So, so it seems like this is cross-sectional data, not time series data, so we can't actually tell that patients are moving in exactly this way, but we have other time series data in 2 drug combination therapies that does suggest actually that patients are moving from one category to another, first getting single drug therapy and then getting double drug therapy. And it turns out that it's not just this particular therapy. 3 drug combinations fail in a partially predictable order across many different types of therapies. So one of the things we did is we went back to that data set of nearly 7,000 patients that I showed you previously, and we looked at patients who were only given 3 drug combination therapies that sort of fell into treatment regimens that looked something like this. So they had a 3TC or FTC, which is a specific type of NRTI, which is really common. They had, a different NRTI, and they had protease inhibitors. And we subsetted out the patients that had exactly 1 drug resistance mutation that we could spot within reverse transcriptase or within protease. And what we see is that overwhelmingly in these different types of therapies, 3TC resistance is the first drug that, or 3TC is the first drug to which resistance emerges. You can also look at these NNRTI-based therapies, which is just a different class of drugs, and we see that overwhelmingly NNRTIs are the first drugs that fail when treated with these particular combinations of drugs. So this presents a puzzle. We've now observed a couple of different things that don't seem to fall at all within this textbook idea of how drug resistance emerges within HIV in the context of triple combination therapies. So to sort of summarize the weird observations I've just told you, first, drug resistance still does emerge under combination therapy. It does so via single drug resistance mutations, and it does so in what we believe is a partially predictable order across a wide variety of different drugs. So how can we explain these sort of 3 weird different observations? So previously in all of the models, or the sort of internal picture of how I think about HIV and treating HIV is that within a person you have this sort of big bag of viruses, and you treat them with a combination of a bunch of different drugs, and the drugs affect all of the viruses. In reality, there's sort of evidence that HIV is not a single well-mixed population, but actually is a series of subdivided populations interconnected by some amount of migration. If that's the case, then it's possible that not every drug is getting to every part of the body equally, that you have certain parts of the body in which certain drugs are able to reach better, and other parts of the body that have less drug that are able to reach this, to reach those particular parts of the body. Now, this is sort of something that people have been thinking about within the context of HIV for a while, and there's this really nice modeling paper that came out in PNAS a few years ago concerning just what we think that this would do to the dynamics of HIV drug resistance evolution. And what I want to argue is that I think that this type of a model does a really good job of explaining the sort of weird observations that we've observed in clinical data. So if we think back to that example that I showed you previously, where we have 3TC, NFE, and D4T, and we see that patients are acquiring drug resistance in this sort of 1 at a time, partially predictable order, we can think about what are the penetrances of these drugs to different parts of the body and see whether this makes any, helps us explain the data at all. So we know that both 3TC, NFE, and D4T, all 3 of these drugs are relatively good at penetrating to most places in the body. D4T is a little bit of an exception just because it has a very short half-life, and so it's very possible that this drug is sort of effectively getting to the body in a it's sort of not as strong a drug, both in terms of it's how long it's sticking around the body, but also just in terms of what is the effect of this particular drug on the virus, even in the absence of these sort of spatial temporal concerns. What sort of gets more interesting is thinking about how these drugs penetrate into the central nervous system. So we know that 3TC is quite good at getting into the central nervous system, but nalfinavir, NFE, is actually very bad at getting into the central nervous system. We think that this is very much in line with the patterns that we're seeing here. Namely, there is a part of the body in which, mostly only 3TC is the only drug that's present at a particular point in time. And so the viral population as a whole can acquire resistance to 3TC. We know that this works relatively well in terms of viruses that are able to acquire resistance to monotherapy. And then this provides a sort of sanctuary part of the population where HIV is able to evolve to sort of a much simpler evolutionary task than the thing that we think we're putting forward. then the virus can go to different parts of the body and evolve to different hypothetically to different drugs. So what would happen in this case if we took NFV and we replaced it with boosted lopinavir, which is a more modern version of the class of drugs that NFV is. Now, lopinavir does better, although not as good as 3TC at getting into the brain. So what we expect to happen in that case is that so the question is, what actually happens in the data if we look at patients who were treated with this combination therapy? And what we see is that, sure enough, there are a lot of patients that don't have any drug resistance in this case. And there are some patients that do acquire 3TC resistance, but there's no ongoing evolution beyond the single drug resistance. So what this points to is this idea that lopinavir is actually protecting 3TC. It's a little bit of a counterintuitive result, because by adding lopinavir, what you're doing is effectively shielding 3TC from being in the single drug compartment where evolution of resistance to just 3TC is possible to happen. So one interesting side effect of this is thinking about, if you're designing therapies, it doesn't matter just sort of where your worst drugs are getting, but it actually matters where your best drugs are getting. Because if you have single drugs that are getting to certain parts of the body much better than the other drugs that you're using in this combination therapy, this can actually be a liability in terms of allowing spaces where HIV is able to evolve resistance. So what I've shown you so far is sort of anecdotal. It's really difficult to get all of this information about the drug penetrances to different parts of the body. This is something that we're currently working on, and we sort of have this ongoing project where we're trying to add in some dynamical modeling to try to figure out whether there are other explanations for these types of patterns, including weird temporal adherence patterns that can allow specific parts of the body, to allow the body as a whole to have different drug levels. So that's work that I hope is coming out somewhat soon. But in general, this is sort of an idea that we're really intrigued by. How can we understand this combination therapy failure, and how can we explain these weird patterns that we're observing in clinical data? So one reason that we think that HIV might be able to do this is because it's actually exploiting heterogeneity, and that these complex simultaneous challenges are not quite as complex and simultaneous as we might think that they are. So there's sort of two things that we have left to nail down here. One of them is how are the drugs distributed? If drugs aren't getting to different parts of the body at different levels, then this hypothesis doesn't make a lot of sense probably. But we have really ample evidence that the drug distributions are pretty variable throughout the body. The question is just nailing them down in a sufficiently quantitative way that we can actually make predictions off of it. The other question that we need to consider is that the viral distribution in different parts of the body can't be too homogeneous. So if it's really the case that every virus is sort of experiencing all of the different environments of all of the different parts of the body over very short timescales, it probably doesn't make sense to think about this as a suitable hypothesis, in part because it means that the virus is feeling an average of all of these different environments, and that being for some period of time in a place where a single drug resistance mutation is beneficial doesn't have nearly as strong an effect as if the virus and its descendants are staying in a particular place for long periods of time. So in this final part of the talk, I just want to dive into this viral distribution question a little bit. Where are viruses getting to different parts of the body? How are they evolving in those different places? And how connected are different body parts? And I think that sort of nailing down this side of the question is sort of a necessary step before we're able to consider the spatial monotherapy hypothesis in as much detail as I would like. So this is a question that I initially went back to the literature in order to try to answer. Does the HIV population within a body look well mixed? And I found many studies that had assayed viral populations from different parts of the body, maybe the blood and the lymph node, or the blood and the genital tracts, or maybe the blood in the gut. And I found many studies that had tried to ask these questions and categorize whether HIV within a patient does or does not look well mixed. But when I tried to categorize these different studies based on their results, I found that the answers were pretty mixed, that some studies found really strong support for viral compartmentalization, and some studies found no evidence at all for viral compartmentalization. So what I was left with was kind of scratching my head. How can we explain not only whether HIV is moving around the body fast enough to allow this spatial monotherapy hypothesis to be plausible, but also how can we explain all of these different studies that have found such drastically different results? So to do this, I wanted to sort of step back and ask this question about how are these strong evolutionary forces playing out over their spatial contexts? So within the context of a single patient, what does viral evolution look like in these different parts of the body? And I really want to focus on doing this quantitatively, trying to put some parameter estimates on these different forces, in part because I want to sort of model the spatial monotherapy hypothesis, and nailing down what migration looks like here is going to be a really key part of that. So to do this, I'm going to kind of show you a couple of different threads that are leading into this, looking at what these strong evolutionary forces look like just via observation by sampling in different parts of the body, by doing a little bit of modeling, and then finally combining these together to do some inference on what these parameters actually could be. So as I was pondering this question, I was really lucky to have formed this collaboration with Zandria Ambrose at the University of Pittsburgh. So Zandria is an HIV biologist who works in a variety of model systems to study HIV, well, both in the lab, but also in experimental populations. And so before I had ever met her, she had done this experiment in which she had infected macaques with simian HIV, and watched how this, had infected macaques with simian HIV, had treated these macaques, and then had watched how the virus had spread to different parts of the body and evolved drug resistance over time. So simian HIV is an SIV, so a simian immunodeficiency virus that has an HIV reverse transcriptase spliced in. What this means is that you can treat SHIV with HIV drugs, and they get the exact same types of resistance mutations that HIV does, which is a really convenient model system. So what Zandria did is she infected these macaques with RT-SHIV, and then had allowed the viral population to establish within the macaque, and then had treated these macaques with a monotherapy, which had initially succeeded, but ultimately failed in suppressing the viral population. So she had treated so monotherapy is not exactly what I'm interested in, but it turns out that by treating these macaques with monotherapy as opposed to these much more complex therapies, you're able to get some gauge on what these evolutionary processes look like with a much higher probability. Of course, if this is a relatively rare event, accumulating resistance to combination therapies, it's much harder to observe an instance in which this is happening within a model system. But here, treating with monotherapy is basically guaranteed to evoke drug resistance evolution within short timescales. So Zandria sequenced or sampled the population before, during, and after the evolution of drug resistance, but then also excitingly, she has samples from multiple different spatial locations from within the macaque. So she has plasma, lymph node, and gut samples taken at each of these different temporal time points. And at each of these time points and at each of these locations, she has approximately 30 single genome sequences from which she can we can try to figure out what's going on within the population. So I'm going to show you the data from these various different compartments to just sort of give you a sense of what these evolutionary trajectories look like. And when you look at the blood plasma, what you see is as follows. So this is sort of a semi-Muller diagram with a little bit less resolution because there isn't enough genetic diversity within the sequenced region, which is just the pol gene, to get a sense of everything that's going on across the viral lineage. But what we see here is that initially the population is entirely wild type, and very quickly, multiple different drug resistance mutations take over. So anything that is underlined here is a viral drug resistance mutation. Seeing this picture, it sort of gives us some sense of what are the strengths of the genetic parameters that are at play here. Namely, we see that this is a soft sweep. We see M184V and M184I sweeping simultaneously, and also M184V sweeping with the synonymous mutation. It's unclear whether this happens before or after, whether these are actually two separate events. And we see that it happens really quickly. So evolution is happening on the same sort of timescales that we would have predicted happened under monotherapy previously. Now, if you look across to the lymph node and the gut, you can see that sort of similar things are playing out, but not exactly the same. Namely, you see mostly the same drug-resistant variants spreading in each of these different compartments. The gut is a little bit of an exception here, but they're not doing exactly the same thing. So you can look at the trajectory of M184V here, which is initially rare and then becomes more common, whereas in the gut, you see that M184V is initially common and becomes increasingly rare. So from these pictures, it's clear that migration is sufficiently strong to mix these different variants. However, it's not so strong that it's making the trajectories look exactly the same in each of these different compartments. So what we can see from these pictures is that mutation, selection, and migration are all apparently strong, but it's sort of unclear how strong they are relative to each other. So in order to get a handle quantitatively on how strong these different parameters could be relative to each other, I go back to sort of the model that we started with, this very simple Wright-Fisher model with selection, and just look at this sort of only slightly added complication that we're thinking about how this might look across multiple different spatial compartments. So here I've just illustrated 2 of them. So now we have independent processes playing out in these 2 different spatial compartments, but they're connected by migration. So as an additional step each generation, some number of each virus has a probability m of migrating to the other place. And we consider what's happening even if these populations are experiencing exactly the same selective pressure and they're getting mutations either red in population A or blue in population B that are effectively entirely equivalent. Then what we can do is, what are the dynamics of these different population parameters that need to happen in order for us to get population patterns that look something like what is happening within our SHIV patients or SHIV experiments. And so what we can do is look at these trajectories at different migration rates and sort of try to titrate the migration rate until we find one that looks something like what we're observing in our actual genetic data. And so if you let the migration rate be really small, what you see is that each population independently fixes its own variant. So population B fixes a blue variant, population A fixes a red variant. And you can kind of see this in the FST trace that, initially the populations are identical and then over time they diverge. And even as you raise the migration rate to higher and higher levels, you see that this pattern persists. So even with quite high migration rate, NM equals 10, which in sort of a neutral context is actually a relatively high migration rate, we see that over these very short timescales of evolution, we see that there are sweeps happening independently in both compartments and sort of FST is going up and staying up. I should mention here that I'm using quite strong selection strengths and population mutation rates. I think theta here is 1, maybe 0.5. And the selection strength is also 1, so it's sort of a 100% advantage of the resistant type versus the sensitive type. So under these strong popgen regimes, it seems like even quite a large amount of migration doesn't do anything to equilibrate the populations. Now, on the other hand, you can crank migration up extremely high. And if you have the migration rate be sufficiently high, you see that the trajectories look effectively identical across the 2 compartments and FST is never elevated. Now, where it gets interesting is sort of in this intermediate regime in which you see that the population migration rate is somewhere in this sort of 100 to 1,000 regime. And what we see is that the same types of patterns that we noticed in the SHIV data are happening here as well. Namely, initially, population A is dominated by a particular type of variant, which equilibrates into population B-like variants over time. And population B is initially highest frequency in its own local variants, and over time, it's getting increasing numbers of population A variants. So what this is suggestive is that there are particular migration rates that can kind of contribute to these 2 different parameters, these sort of patterns that we see that are similar and SHIV-like. We did a little bit of work on trying to nail this down analytically, which I'm going to skip because what I want to say is we can use some of this intuition in order to try to get a sense of how to actually get popgen parameters out from our SHIV data. And so what we did, I know that at IE2, or maybe just I'm thinking mostly in the Kern-Ralph collab in particular, you have a lot of fancy machine learning to do estimation techniques or to do estimation, but we did something much simpler than that. We just did ABC, in which we took priors over these various different distributions, for example, theta, S, and M. We chose a particular value of those different distributions. We use those to simulate trajectories under those particular population parameters. And then we can summarize these statistics, these time series in various ways using a variety of statistics. And then what we can do is we can compare this to the SHIV data in which we can compute these parameters directly from the data. And in doing so, if these 2 things are sufficiently close together, we can effectively choose to accept or reject this particular set of parameters. And if it's sufficiently close, you can add theta, S, and M to this posterior. And then you can do this over and over again, sampling from the priors, simulating under it, summarizing the data, and then comparing it in order to generate these posteriors, which can give us a sense of what are the relative values of theta, S, and M that can produce the type of data that we're seeing. And when you do this, you can actually get a sense from the SHIV data, what are the relative parameter values. And I'm going to be showing you pairwise comparisons of these different compartments. So if you're estimating, for example, the selection strength, the migration rate, and theta in these different compartment pairs, what do you see? And so there's not a particular reason that you couldn't do this with higher order, doing this a three-way instead of a two-way comparison. But because we think a lot of the interactions between HIV and different parts of the body are going to be mediated via the plasma is sort of a good example case of migrating into and out of this compartment in order to say something about these parameters. So if you do this and you try to fit the selection strengths, you get between the plasma and gut somewhere between 1 and 10 as our estimated selection strength. You can do something similar between the plasma and the lymph node, and you get very similar estimates of how strong selection is. You can do similar estimation and look at the population mutation rate, and similarly, you see that theta is estimated between 0.1 and 1. Now, I'll emphasize that these are sort of crazy parameters if you're thinking about human populations, but in the context of the time series that we're seeing in HIV, they actually kind of make a lot of sense. And actually, if you take those parameters and you map them back to this plot I was showing you at the beginning, this is exactly the parameter space where we would expect to have often sweeps in 30 generations and highly soft sweeps. Now, where things got a little bit more interesting when we actually went to estimate the migration probabilities. So this is the migration probability between the plasma and the gut, and this is between the plasma and the lymph node. And what we can see is that there's a lot more evidence for faster migration between the plasma and the lymph node than the plasma and the gut, suggesting that these are much more interconnected compartments than the plasma and the gut are. Then, so I was sort of excited about this because if you look at this NEM equals 1 point, which is generally what we consider the place where it's easy to estimate migration rates from this sort of this canonical right formula under the sort of neutral equilibrium, we think that we shouldn't really be able to estimate migration rates that are much higher than this mark. But because we're looking at dynamical data that is very much not in equilibrium and is experiencing these really fast, strong selective sweeps, by looking at how quickly the population is thrown out of equilibrium and then sort of recalibrates to equilibrium, we can say something about what these much faster rates are. It's sort of like using selective sweeps as your measuring stick to understand how fast these forces are instead of using genetic drift. So these different migration rates actually suggest pretty different residence times in these different compartments, namely that populations are not spending very much time in the lymph node before they're migrating back out into the plasma, whereas they're spending considerably longer in the gut, which really does suggest that if there are different amounts of drug getting to different parts of the body, that this is really going to drastically, that if populations staying in the gut for long periods of time may really be able to feel these differences and hypothetically evolve to follow this sort of optimum, which might be different in the gut than in the lymph node. So how do these strong evolutionary forces play out over space? They play out quickly and dynamically with the potential for estimating these very fast migration rates because you're comparing the strength of this fast migration to these relatively strong forces of selection. But there's a lot more that we want to do here. There are sort of, we could imagine that with better data, we could fit much more exact parameters. And in fact, it might be fun to have some conversations with other folks at IE2 about what are the other ways we could do better estimation and incorporate some of the newer techniques into this question. But I kind of want to circle back to this idea that rapid adaptation is extremely widespread. And though I've only been talking to you about HIV for the past 45 minutes or so, check the time, that these are actually populations that demand to be understood in greater detail. And the sort of short-term dynamism that we're seeing in these populations can often be averaged out if you're trying to look at what is happening in the genetics of these populations over long timescales. So looking at this data with a resolution that is both temporal and ideally spatial can help us get a sense of what these population parameters are, how they evolve, and how we need to understand these in a way that's sort of, in some ways, fundamentally different than what we're thinking about in how much slower evolving populations evolve. So I want to make sure there's at least a few minutes for questions. So I want to sort of close by saying that I hope that I've illustrated that at least in HIV and hope what I believe is in a wide variety of other natural populations, extensive genetic variation fundamentally alters how evolution proceeds, and specifically how this evolution proceeds in both space and time. And I think that using existing medical and agricultural data, we're poised to make really fast progress in understanding some of these important questions. So with that, I want to thank my collaborators and co-authors on all of these projects, particularly Pluni Pennings and Dmitry Petrov. My co-authors on all of these papers, Sandra Ambrose is the sort of person who is behind the SHIV collaboration, as well as funding that supported me during this time. Members of the labs that have always been so amazing for conversations. And before I take questions, I do want to mention, as Vince alluded to, that I am opening a lab in June of this year in Seattle at the University of Washington in the Genome Sciences Department. And I'm actively looking for graduate students and postdocs, although I think that the graduate school deadline is already closed for this year. But if you feel like reaching out and having some sort of even informal conversations about this type of work and other types of pieces of work that we're thinking about moving forward, even very casual interests or just feel free to reach out. I'm always excited to have those conversations, especially if during your time in Oregon, you've fallen in love with the Pacific Northwest. It is a mere 102-mile stroll to get to Seattle, so not too far. Finally, yeah, I'm happy to take any questions about that or anything. Thanks very much for your attention.
Thank you so much. That was such a fascinating talk and extremely thought-provoking. I'm happy to take questions if folks want to type them in the chat, or let's see, is there a way to see everyone simultaneously?
They can just unmute themselves also.
Yeah. Okay, so we have a question in the chat from Monica. Is the drug resistance mutation also causing more severe symptoms or more virulent HIV, or are these trait mutations independent of each other?
Yeah, that's a great question. So we think that it's actually the opposite. I think that they're largely independent, and if anything, that there's some evidence that these drug resistance mutations are not affecting, for example, transmission or virulence. So one of the reasons that we believe this is because there's a relatively low rate of transmitted drug resistance, or at least there was until relatively recently, and that there's this global worldwide pattern where HIV sort of intra-patient evolution, if you have someone who is infected and then evolves drug resistance or whatever, they're much more likely to transmit something that is non-drug resistant from patient to patient. And it could be that it's possible that these particular mutations I mean, we know that they're associated with fitness costs. And so we don't think that they're really passed between patients very often. In terms of virulence, I mean, I think the major effect that they have is that it means the drugs stop working. And so the population sizes are able to grow again. And that, of course, is bad for patients. But if you switch them to other drugs, the drug resistance mutations are quickly lost, because we think that they come with pretty substantial fitness disadvantage. Their fitness costs is the word, yeah. And then I think you mentioned that is it associated with virulence, or what was the other half of the question?
Yeah, more severe symptoms or more virulent HIV, or are these trait mutations independent of each other? Yeah.
Yeah. I think that they're it's largely fair to think that they are not increasing virulence. They're not increasing transmissibility. They're mostly just hurting patients because they mean that the drugs stop working.
I had a question, but it's too hard to type. So how do you think that standard of care plays into this? So as far as, do you have information in your data as far as like, are the combinations of drugs typical as far as like, are they usually given all in combination all at once at different doses? I imagine like that could play into how patients respond. And is it possible that different combinations of drugs could sort of force the virus into sort of an evolutionary trajectory where it would maybe be more vulnerable or less vulnerable, if that makes any sense?
Yeah. So in answer to your first question, so one of the sort of unresolved mysteries here is, or I guess maybe a question that I didn't answer directly within the talk, was sort of why exactly are these lines going down if there are triple therapies that are, that if triple therapies have been used from here to here. And I think one of the answers does have to get to do with your first question, which is how hard are these drugs to take? What are the associated symptoms? So if is someone going to be less likely to actually be adherent to their therapy if there are terrible, nausea associated? And I think the answer to that is yes. So many of the drugs are better. One of the reasons that we think drug resistance has gone down is because drugs are better tolerated by people. Like, we found ones that have less associated toxicity and those that have simplified regimens. So there are certain drugs that have half-lives that are so short that you need to take them 3 times a day, which is hugely burdensome. Now, I know that there are at least 2 drug pills. So one, I guess PrEP is one of them, things where you take multiple drugs within the same pill. I don't know whether there are 3 drug ones. I suspect that there are, but I don't know that for certain. But it's certainly the case that the easier it is that we make it for people to take drugs, the more adherent they are. And we know that there's this sort of relationship between adherence and how many drugs or adherence and probability of resistance. So if you're taking all of your drugs 100% of the time, you have a relatively low probability of evolving resistance. If you don't take drugs ever, you also have a relatively low probability of evolving drug resistance, simply because you're not creating the evolutionary pressure for the virus. But you're right, definitely, that adherence kind of plays in here, and by these better drugs are in part better because they make it easier for people to adhere.
Well, I was actually getting at sort of order, essentially. So if there's information on if you treat with one drug and within a period of time another drug and then another drug, sort of, can you sort of within a temporal period of not allowing sort of enough? Am I making any sense?
Yeah. So you're talking about like pulse dosing.
Yeah. Yes.
So yeah,
is that
can you set up
this data, or is it sort of?
I don't know directly. I suspect that this is something that because of this toxicity question, there were experiments with, for example, taking people off drugs for drug holidays for periods of time. I don't know about particular experiments with putting people on one drug and then a second drug and then a third drug, in part because if you can take all 3 drugs simultaneously, it seems like it's strictly better to do that, simply because you're not creating these spaces or these times where HIV is able to solve tasks that are evolutionarily simpler. So I don't know whether there's been a lot of research into that, although I do know that this is something that people are really curious about with respect to antibiotics. I think with HIV, the idea is just more is always better, provided toxicity is not too much.
Right. I guess I was getting at, can we break these into, you're pointing out, evolutionarily simpler problems, and then sort of essentially, is there an evolutionary trajectory that is predictable for drug 1, drug 2, drug 3, and then can we, from that, wrap our minds around some type of order that might be more efficient? But that's kind of a complicated, weird question. Sorry.
No, it's okay. But I think the answer to it is that with HIV in particular, it just seems to make sense. The lowest rates of evolution are by treating everything together, and ideally getting everything in perfect synchrony, both temporally and spatially. If you could get every drug to every part of the body, that would be ideal. And that by kind of separating these things out in time, you're likely creating more opportunities for the virus to be facing something simpler.
So I think we should take more questions during the tea time. So we can all stay in the same Zoom meeting, but let's maybe take 5 minutes. But first, let's thank Alison for a wonderful talk and for this great virtual visit.
Yeah, and thank you. It's been really fun. I've really enjoyed the conversations that I've had with folks, and I hope that